Welcome to our deep dive today. We're going to be talking about Actinobacillus actinomycetemcomitans.
Ooh, that's a mouthful.
It is, and thankfully it's often shortened to just AA.
Okay, good.
But yeah, we're gonna be talking about this really fascinating bacterium that is a major player in periodontal disease.
Right.
And we are sourcing our information from Principles and Practice of Clinical Bacteriology.
A great book.
Yeah, it's a great resource, and I think this will be a particularly interesting deep dive for medical microbiologists.
Absolutely.
You know, who are really familiar with the oral microbiome,
Right.
and all the different players and how they interact and sometimes cause trouble. So let's dive right in. Can you just give us a brief overview of Actinomycetemcomitans? What is it? Where do we find it?
Yeah, so Actinomycetemcomitans is a small gram-negative rod.
Okay.
And it's actually a normal resident of the oral microbiota.
Oh, interesting. So we all have this potentially pathogenic bacteria just hanging out in our mouths.
Yep, that's right. But it's interesting because despite being part of the normal flora, it's also heavily implicated in localized aggressive periodontitis.
Okay, so it's kind of a Jekyll and Hyde situation.
Exactly.
It can be harmless, but under certain circumstances, it can turn nasty.
Yep.
Before we get into the pathogenic side of things, I'm curious about the name Actinobacillus actinomycetemcomitans.
Yeah, it's a mouthful.
It is.
It's a long one.
Yeah.
And it kind of reflects the evolving understanding of this bacterium. So initially it was thought to be linked to actinomycosis.
Hence the actinobacillus part.
Exactly.
Okay.
But later research showed that it's actually quite distinct from the true actinomycetes.
Interesting.
But the name kind of stuck. And it even went through a phase as Haemophilus actinomycetemcomitans,
Oh, wow.
highlighting its early association with the Haemophilus genus.
So it's had a bit of an identity crisis over the years.
It has. But today it's firmly placed within the genus Actinobacillus based on its genetic and biochemical characteristics.
Okay, so Actinobacillus it is.
Yep.
What about its appearance? What does it look like under the microscope?
So it's a small, non-motile gram-negative rod, as I mentioned.
Okay.
And when you culture it, it forms these really distinctive, rough, star-shaped colonies.
Star-shaped.
Yes, star-shaped. It's actually quite helpful for initial identification in the lab.
So if you see those star-shaped colonies on a plate, you think, could be 80.
Exactly.
Interesting. What else is important to know about its physiology or its kind of lifestyle?
One key feature is its facultative anaerobic nature.
Okay, can you explain what that means?
Sure. So it means it can thrive in both aerobic and anaerobic environments.
That makes sense considering it lives in the mouth where conditions can vary.
Exactly.
You've got pockets where there's less oxygen.
Right.
You've got surfaces that are exposed to air.
So this adaptability is crucial for its survival.
And unfortunately for its ability to cause disease.
Yes, unfortunately.
Let's talk about that. What makes this seemingly harmless resident of the oral microbiome turn into a troublemaker?
Well, ADA has a few key virulence factors that make it pathogenic.
Okay, let's hear about those. What's the first one that stands out?
Well, the first one is its ability to adhere to surfaces.
So it needs to stick around to cause trouble.
Exactly. And it does this through a special set of genes called the TAD locus.
TAD locus.
Yeah.
What does that stand for?
It stands for tight adherence.
Okay, that makes sense.
And it allows ADA to produce these hair-like structures called pili.
Healy.
Yeah.
Okay, I've heard of those. They're like little grappling hooks that bacteria use to attach to things.
Precisely. And in the case of AA, these pili allow it to cling tenaciously to teeth, gums, and other surfaces in the mouth.
So it gets a good foothold, and then it can start causing problems.
Exactly. And what's even more fascinating about the TAD locus is that it's not just limited to AA.
Okay.
It's considered a widespread colonization island found in other bacteria and even some archaea.
Archaea. Wow. So this adhesion mechanism is ancient and widespread.
It is. It suggests that adhesion is a fundamental requirement for bacterial survival and colonization regardless of the niche.
That's incredible. So archaea gets a foothold using its tight adherence pili. And then what? How does it actually cause damage?
Well, once it's attached, it can start deploying its toxins.
Toxins. Okay, this is where things start to get really interesting.
It is. AB produces two key toxins that contribute to its virulence.
Okay, let's break those down one by one. What's the first toxin?
The first one is called leukotoxin, or LTXA.
Leukotoxin.
And it's particularly nasty because it specifically targets white blood cells.
White blood cells. Those are the cells that are supposed to protect us from infection?
Exactly. And LTXA targets neutrophils and monocytes, which are key players in the immune system's defense.
So it's not only infecting the host, but also weakening their ability to fight back.
Precisely. And to make matters worse, there's a particularly aggressive strain of AA called the JP2 clone,
JP2.
which produces significantly higher amounts of leukotoxin.
Oh no. So this JP2 clone is like the supervillain of AA.
It kind of is. And this increased production is due to a deletion in the promoter region of the gene that controls leukotoxin production.
So it's like they've removed the brakes on leukotoxin production.
Exactly.
That's scary. Okay, so we got leukotoxin. What about the second toxin?
The second toxin is called cytolethal distending toxin, or CDT.
CDT. And how does this one work?
It disrupts the cell cycle, preventing cells from dividing properly and eventually leading to cell death.
So it's like a cellular time bomb.
Pretty much. And this can cause significant damage to host tissues and further cripple the immune response.
Two different toxins. Two different ways of wreaking havoc.
That's right. But the story doesn't end there. ARIA also has a range of other virulence factors that contribute to its pathogenicity.
Like what? Give me an example.
Well, one example is chaperonin 60.
Chaperonin 60.
Yeah.
What does that do?
So normally, chaperonin 60 is a good guy.
Okay.
It helps proteins fold correctly within cells. But in AA, it's also found on the cell surface where it can interact with host cells and trigger harmful inflammatory responses.
So it's like it's taken this helpful molecule and turned it into a weapon.
Exactly.
That's fascinating and a little bit terrifying.
It is. And then there's the most intriguing virulence factor of all.
Okay, I'm all ears.
Cellular invasion.
Invasion. So AA can actually get inside host cells.
Yes. Unlike many bacteria that remain outside host cells, AA can invade non-phagocytic cells, including epithelial cells in the mouth.
Wow, so it's like a microscopic Trojan horse.
Yeah.
Sneaking into cells and setting up shop.
Exactly. And once it's inside, it can replicate much faster than it does outside.
Oh no, so it's using these host cells as both a shield and a breeding ground.
Precisely. And it can even move from one cell to another, spreading the infection while remaining hidden from the host's immune system.
So it's like a master of stealth and sabotage.
It really is.
This is all pretty impressive from a bacterial standpoint, but I imagine it's pretty terrifying from a patient's perspective.
It is, and we'll talk more about the clinical implications of these virulence factors in the next part of our deep dive.
Sounds good. I'm looking forward to delving deeper into the world of Actinobacillus and learning more about how we diagnose and treat these infections.
Me too. It's a fascinating bacterium with a lot of surprises. So let's talk about what happens when A starts causing problems in the mouth.
Yeah, this is where things get real for patients.
Exactly. As we mentioned earlier, AA is heavily implicated in localized aggressive periodontitis, or LAP.
LAP. Okay, and just to remind our listeners, what's the hallmark of LAP?
LAP is characterized by rapid bone loss.
Okay.
And this bone loss is often localized around the incisors and premolars.
So those front teeth?
Yes, exactly. And this can eventually lead to tooth loss if it's not treated.
And you mentioned that LAP tends to affect younger individuals.
That's right. It typically strikes people under the age of 30. And it can progress very quickly. So early diagnosis and treatment are really important.
That's concerning. So it's not just any old gum disease.
No, it's a specific and aggressive form that requires prompt attention.
Before we delve deeper into the clinical aspects, let's talk about how we actually diagnose an AA infection in the lab.
Okay, sure.
I imagine it can be tricky considering all the other bacteria that live in the mouth.
You're right. It does require careful techniques to isolate and identify AA from the rest of the oral microbiota.
So where do you start?
Well, we typically start with a sample, which could be dental plaque, a tissue biopsy, or even an aspirate from an infected site.
Okay.
And the key is to collect the sample as cleanly as possible.
I can see how that would be challenging. There's so many different species in the mouth.
Exactly. We want to minimize contamination from other oral microorganisms.
You've got your sample. What's next?
The next step is to culture the sample in the lab.
Okay.
Now, AA is a bit finicky.
Oh,
It prefers a CO2-rich environment, and it grows best on blood or chocolate agar.
Okay, so you need to give it the right conditions to grow.
Exactly. And remember those characteristic star-shaped colonies we talked about?
Ah, yes, those are a good clue.
They are. When you see those on a culture plate, it's a good indication that you might be dealing with AA.
But you can't rely on appearance alone, right?
Right. We need to confirm the identification using biochemical tests.
Okay, like what?
Well, we use tests like the catalase and nitrate reduction tests. These help differentiate AA from other bacteria that might have similar colony morphology.
So you're looking for a specific set of biochemical reactions.
Exactly. And of course, nowadays we also have molecular methods like PCR.
Ah, yes, PCR, the gold standard for bacterial identification.
It is. We can use PCR to amplify and sequence the 16S rRNA gene, which allows us to identify the species with a high degree of accuracy.
And you can even identify specific strains like that highly virulent JP2 clone.
Exactly. So we've got a lot of tools at our disposal to accurately diagnose AA infections.
That's great. So we've talked about the bacterium itself, its virulence factors, how we diagnose it in the lab. Now let's shift gears and talk about prevention and control.
Okay, sure.
How do we keep this bacterium in check?
Well, the first line of defense is good oral hygiene.
So brushing, flossing, the usual advice.
Exactly.
Wow.
Regular brushing and flossing are essential for removing plaque, which is that sticky film of bacteria that forms on teeth.
Right.
Plaque buildup creates the perfect environment for AA and other bacteria to thrive.
And I imagine regular dental visits are important too.
They are. Professional cleanings can remove plaque and calculus, which is hardened plaque that's harder to remove at home.
Okay.
And these cleanings also give the dentist a chance to assess your overall oral health and look for any early signs of periodontal disease.
So good oral hygiene is key. But are there any other preventative measures that might be helpful?
In addition to mechanical cleaning, antimicrobial mouthwashes can also be effective.
Okay.
Chlorhexidine in particular has been shown to be quite effective against AA.
Interesting. So it's a combination of mechanical removal and chemical warfare.
You could say that, but what happens when those preventative measures aren't enough and someone develops a full-blown AA infection?
Yeah, what are our treatment options then?
Well, the treatment approach depends on the severity and type of infection.
Okay.
For LAP, the goal is to halt the progression of bone loss and save the affected teeth.
Right.
And this typically involves a combination of mechanical debridement and antibiotic therapy.
Can you explain those a bit more?
Sure. Mechanical debridement is essentially a deep cleaning procedure.
Okay.
It involves removing plaque and calculus from below the gum line where AA likes to hide.
So it's like a really thorough cleaning of the infected areas.
Exactly. And along with the mechanical cleaning, we often prescribe antibiotics to target the bacteria directly.
What kind of antibiotics?
Tetracyclines are the go-to antibiotics for AA infections.
Tetracyclines.
They have good activity against this bacterium, and they penetrate well into the gum tissues.
I've heard that tetracyclines are commonly used for periodontal disease.
They are.
Are there any other antibiotics that you might use?
Yes, sometimes we might combine tetracyclines with other antibiotics like metronidazole or amoxicillin. This helps broaden the spectrum of coverage, especially if we suspect a mixed infection or if the tetracycline alone isn't working.
So it's like hitting the bacteria from multiple angles.
Exactly. But I have to say some infections can be quite persistent or recurrent even with appropriate treatment.
Oh, that's frustrating.
It is. And those cases often require a more multifaceted approach.
Like what?
We might consider using things like bone replacement materials, growth factors, or even cytokine inhibitors to modulate the inflammatory response.
So it's not just about killing the bacteria, it's also about helping the body heal and repair the damage.
Exactly. It's a holistic approach.
Right.
And just as important is the ongoing management of these infections.
Right. So once the acute phase is under control, what happens next?
Well, long-term management really comes down to two things: patient education and addressing underlying risk factors.
Okay.
We need to empower patients to take an active role in their oral health.
So it's about more than just telling them to brush and floss.
It is. We need to help them understand the disease process, the role of AA, and the importance of consistent oral hygiene.
Right.
And we also need to address any underlying risk factors that might make them more susceptible to infection.
Like smoking.
Exactly. Smoking is a major risk factor for periodontal disease. It impairs blood flow to the gums, weakens the immune response, and creates an environment where bacteria like A can flourish.
So it's really a team effort between the healthcare provider and the patient.
Absolutely. Open communication, patient education, and a proactive approach to managing risk factors are all crucial for long-term success in controlling AA infections.
I think we've covered a lot of ground today. We've talked about AA's virulence factors, how we diagnose it in the lab, and the different approaches to prevention and treatment. Now, to wrap things up, let's test your knowledge with a few exam questions.
Sounds good. Let's see what you've learned.
Okay, so let's go back to those exam questions and see how well you did. So the first question was, what are the 4 key virulence factors on A, and how do they contribute to its pathogenicity?
Right. So number one, adhesion.
Okay.
Adhesion. A has that TAD locus,
Right, the tight adherence locus.
which allows it to produce those hair-like pili that help it attach to surfaces.
Are those little grappling hooks?
Exactly. And without that strong adhesion, it wouldn't be able to establish itself and do any damage.
Okay, so adhesion is key. What about number 2?
Number two, toxins.
Toxins.
AB produces two main toxins, leukotoxin and cytolethal distending toxin.
LTXA and CDT.
Yep. And they work in different ways, but both contribute to damaging host tissues and disrupting the immune response.
And don't forget about that JP2 clone.
Right. The JP2 clone produces even more leukotoxin, making it even more virulent.
Okay, so we've got adhesion toxins. What's number 3?
Number 3 is a bit of a grab bag.
Okay.
It includes all those other exported and cell surface virulence factors like chaperonin 60.
All right, chaperonin 60, which is normally a good guy, but AA has kind of weaponized it.
Exactly. And then finally, number 4,
The big one.
cellular invasion.
The Trojan horse.
AA can actually invade non-phagocytic cells.
Right, like epithelial cells in the mouth.
Yep, and it can replicate much faster inside those cells and even move between cells, spreading the infection.
All while hiding from the immune system.
Exactly. So quite a sneaky little bugger.
It is, it is. So to recap, the 4 key virulence factors are adhesion, toxins, exported, and cell surface factors, and cellular invasion.
That's right.
All right, on to question number 2.
Okay.
Which severe form of periodontal disease is strongly associated with AA, and what are its characteristic clinical features?
That would be localized aggressive periodontitis, or LAP.
LAP. And what are the key features?
LAP is characterized by rapid bone loss around the incisors and premolars, those front teeth.
Right.
And this bone loss can lead to tooth mobility and eventually even tooth loss if it's not treated.
And it tends to affect younger people, right?
Yeah. Yes, typically people under the age of 30.
All right, moving on to question number 3. Why are people of African descent more susceptible to LAP?
This is thought to be due to the higher prevalence of the JP2 clone of AA in those populations.
Right, that highly virulent strain that produces a ton of leukotoxin.
Exactly.
Okay, question number 4. What is the significance of the HACE group of bacteria, and which member is AA?
The HAE group is a collection of bacteria that can cause infective endocarditis.
Infective endocarditis, a serious heart valve infection.
Right. And AA is a member of this group, so it's important to be aware of its potential to cause systemic infection.
Okay, good to know. And finally, question number 5. Besides good oral hygiene, what are other important aspects of managing AA infections?
Well, professional dental cleanings are important to remove plaque and calculus. Antimicrobial mouthwashes can also be helpful.
Especially those containing chlorhexidine.
Right, and patient education is crucial.
So they understand the disease and the importance of taking care of their oral health.
Exactly. And of course, we need to address any underlying risk factors like smoking.
Right, smoking is a big no-no for periodontal health.
It is. So it's really a multifaceted approach involving both the healthcare provider and the patient.
A team effort.
Exactly.
Well, I think we've covered just about everything there is to know about Actinobacillus.
We have. From its microscopic features to its virulence factors, its role in periodontal disease, and the different ways to prevent and treat infections.
We even touched on its potential to cause systemic infections.
A reminder that oral health is connected to overall health.
Absolutely. But before we go, I want to leave our listeners with a thought to ponder.
Okay.
We've focused on A as a pathogen.
Right. A bad guy.
But given its ability to invade cells and live inside them, could there be more to the story?
That's an interesting question.
Could A have a role in modulating the oral microbiome in ways we don't yet understand? Could it even be involved in other processes in the body beyond just causing disease?
I don't know, but it's certainly possible. The microbial world is full of surprises.
It is. So to our fellow medical microbiologists, keep those inquisitive minds engaged. Keep exploring, and never underestimate the power of even the smallest microorganisms.
Well said.
Thanks for joining us on this deep dive into the world of Actinobacillus actinomycetemcomitans.